Hello students, welcome back to Cognitive Psychology class. My name is Adwika, and in this video we'll be talking about the topic of cognitive neuroscience. So by discussing about cognitive neuroscience, we actually want to answer these questions. The first of all is, what is cognitive neuroscience, of course, and why it is necessary? And the second one is, how is information transmitted from one place to another in our nervous system? The third one is to answer, how are things in the environment, such as faces and places, are represented in the brain? And lastly, we want to answer what our neural network is and what is their role in cognition. So to answer those questions in this video, we will take up story of cognitive neuroscience. So cognitive neuroscience is the study of physiological basis of cognition. So we will begin discussing the idea of level analysis and then refresh our memory from the biopsychology class about neurons and neural firing. And continuing that, we will take a look at localized and distributed representation in the brain, and finally discuss about neural networks of our brain. So we begin discussing the idea of level of analysis. So level analysis is our rationale behind studying the physiology of the mind, or the cognitive neuroscience, right? And level of analysis here refers to the idea that a topic, one topic, can be studied in a number of different ways, with each approach contributing its own dimension to our understanding, right? That's why we can study the physiology of cognition at levels, ranging from the whole brain, the structure within the brain, to the chemicals created created these structures. So consider, for example, a situation here in which Gil is the male, is talking with Mary in the park, and then a few days later, he passes the park and then remembers what Mary was wearing and what they talk about. This is a simple behavioral description of having an experience and later having a memory about experience. We all have experienced kind of things, right? But what is going on at the physiological level? So during the initial experience in which Gil here perceived Mary as he talking with her, and chemical processes occur in Gil's eyes and ears, which then create electrical signals in neurons, which we will describe shortly. And individual brain structures are activated in Gil's brain, then multiple brain structures are also activated. And all of that leading to Gil's perception of Mary and what is happening as they talk. Meanwhile, other things are happening. So both during Gil's conversation with Mary and after it is over, the electrical signals generated as Jill is talking with Mary will trigger chemicals and electrical processes that result in the storage of Jill's experience. Then, when Jill passes the park a few days later, another sequence of physiological event is triggered that retrieve the information that was stored earlier, which enable him to remember his conversation with Mary. So in the first part, we're talking about Jill's perception, and the second part, we're talking about Jill's memory. So following the idea of that different level of analysis, so one important part of studying cognition is by describing research in cognition at both the behavioral and physiological levels, of course. So we begin our description of physiology by considering one of the basic building blocks of the nervous system, which is the neurons, right? And to understand the relation between the brain and the mind, and specifically to understand the physiological basis for everything we perceive, everything that we remember and we think, it is necessary to look within the brain and observe the small units called neurons that create and transmit information about what we experience and what we know. So this is to refresh our memory, what things that we have studied in biopsychology class, right? Talking about neurons. So for many years, as you may recall from your biopsychological class, the nature of the brain tissues was a mystery at the first, right? And looking at the interior of the brain with the unaided eye give no indication that it is made of billions of smaller units. But then in the 19th century, the anatomists, they applied special stains to the brain tissues, which increased the contrast between different types of tissue within the brain. So when they viewed this stained tissue under a microscope, they saw a network and then they called a nerve net. It was happened in 1870s. The Italian anatomist Camillo Golgi, yeah, he developed a staining technique in which a thin slice of a brain tissue was immersed in a solution of silver nitrate. And this technique then created a picture like the one in this in the figure later I will show you. Meanwhile, the Spanish physiologist Ramon Y. Cajal, yeah, was then using two techniques to investigate the nature of the nerve net. Then, first, he used the Golgi stain, which stained only some of the cells in a slice of brain tissue, and the second one, he decided to study tissue from the brains of newborn animals, because the density of cells in the newborn brain is small compared with the density of the adult brain. So this property of the newborn brain then combined with the fact that Golgi stain affect less than 1% of the neurons made it possible for Kajal to then clearly see that the nerve net was not continuous, but was instead made up of individual units connected together. So Kajal's discovery that individual units called neurons were the basic building blocks of the brain was the centerpiece of the neuron doctrine, or the idea that individual cells transmit signals in the nervous system and that these cells are not continuous with other cells as proposed by the nerve net theory by Golgi. As you can see in this figure, you can see the difference between the A and the B. It's the nerve net theory is in the A figure, yeah, and then the B is the portion of the brain that has been created with Golgi stain, show the shape of few neurons, and there is a neuron cell body, and the thin lines are dendrites or axon. All right, and then to refresh our memory about neuron structure in biopsychology class again, you can see the left-hand figure shows the basic part of neuron, and the cell body is the metabolic center of the neuron, right? It contains mechanism to keep the cell alive, and then the function of dendrites here is that branch, the dendrite is branched out from the cell body is to receive signals, right, from other neurons. Also, there is a axon, also called neuron or nerve fibers, are usually long processes that transmit signals to other neurons. And then the right-hand figure shows a neuron with a receptor that receives stimuli from the environment. For example, this is a pressure stimuli. And those, the neuron has a receiving end and a transmitting end, and its role, as visualized by Kajal in previous study, was to transmit signal. And then continuing Kajal's study of synapses, Adrian in 1920 then further explained the signal travels more clearly using microelectrode. Yeah, so he recorded electric signal from a single sensory neuron using microelectrode, and he found out then when receiving a message from sensory organ or the synaptic, the electrical charge in the neuron had changed, right? And then neurons then able to forwarding a message by regenerate the electrical impulses. So as you can remember that when the nerve is at rest, the membrane cells hold a difference in the electrical charge called the resting potential. It's it's about minus 70 millivolts between the inside and the outside of the axon, right? And as the nerve impulse passes, the charge become more positive, reaching peak and then back at the resting levels. At about the same time Adrian was recording from the single neuron, other researchers also showing that when the signal reaches the synapse at the end of the axon, a chemical called a neurotransmitter is released, and this neurotransmitter makes it possible for the signal to be transmitted across the gap that separates the end of the axon from the dendrite or the cell body of another neuron. And as you can see in this figure, it is the synapses and the neurotransmitter there. All right. Then Adrian also studied the relation between the nerve firing and sensory experience by measuring how the firing of a neuron from a receptor in the skin changed as he applied more pressure to the skin. So the shape and height of the action potential here remained the same as he increased the pressure, but the rate of nerve firing, that is the number of action potential that travel down the axon per second, is increased. All right, so in our previous class, our first class, we have been discussed the mind as a system that creates representation of the world, right? So that we can act within it to achieve our goals. So the key word in this definition is actually the representation, right? Because what it means is that everything we experience is the result of something that stands for that experience. So putting this in neural terms, the principle of neural representation states that everything a person experiences is based on representation in the person's nervous system. To explain that, in the 1960s, researchers began to focusing on recording from single neuron in the primary visual receiving area in our brain. It's the place where signals from the eye first reach the cortex, as you can see in this left-hand side of the brain figure. So the visual cortex here. So the question being asked in this experiment was, what makes this neuron fire? So vision actually dominated early research because stimuli could be easily controlled by creating patterns of light and dark on a screen, and because a lot was already known about vision. But then as research progressed, researchers began recording from neurons in areas outside the primary visual area. And then they found two key facts that many neurons at a higher level of the visual system fire to complex stimuli like geometrical patterns and faces. And the second one, a specific stimulus causes neural firing that is distributed across many areas of the cortex. As you can see in this middle brain figure here, multiple visual area is not only the one part of the visual cortex. And vision, it turns out, isn't created only the primary visual receiving area, but in many different areas as well. And later research extending beyond vision, they found similar result for other cognition. For example, it was discovered that memory is not determined by a single memory area because there are a number of areas involved in creating memories and remembering them later. In short, it became obvious that large area of the brain are involved and creating cognition. As it became clear that understanding neural representation involved casting a wide net across the brain, many researchers began considering the way different areas are connected to one another. So the idea of neural signals transmitted between many destinations in an interconnected brain has led to today's conception of the brain as containing a fast highway system that can be described in terms of neural networks that we will discuss more later. And one possible answer to the question, how can nerve impulses stand for different qualities, is that perhaps there are neurons that fire only to specific qualities of stimuli. So the idea of neurons that respond to specific qualities were brought to the forefront by a series of papers by Hubel and Thorsten. Hubel and Wiesel, is Thorsten Wiesel actually, which would win them the Nobel Prize in 1981. So in the 1960s, Hubel and Wiesel started a series of experiments in which they presented visual stimuli to cats, yeah, as shown in this figure, determine which stimuli cause specific neuron to fire in cat's brain. So they found that each neuron in the visual area of the cortex responded to a specific type of stimulation presented to a small area of the retina. Interesting. And it also shows some of the stimuli that cause neurons in and near the visual cortex to fire, and they call these neurons feature detectors, yeah, because they responded to specific stimulus features such as orientation, movement, and length. Yeah, so as you can see in this different orientation of the bar, it will fire different neurons. So the idea that feature detectors are linked to perception was supported by many different experiments later. And one of these experiments also involved a phenomenon called experience-dependent plasticity, in which the structure of the brain is changed by experience. Yeah. So for example, when a kitten is born, its visual cortex contains features detectors, of course, that is respond to oriented pairs, yeah. And normally the kitten's visual cortex contains neurons that respond to all orientation, ranging from horizontal to slanted to vertical, and etc. And when the kittens grow up into a cat, the cat has neurons that respond to all orientation. But then what would happen if kittens were reared in an environment consisting only of verticals? So Colin Blackmore and Graham Cooper in 1970 decided to answer this question by rearing a kitten in a space in which they saw only vertical black and white stripes on the walls. Poor kittens, right? When a kitten's cortex contained mainly only vertically sensitive neurons, the kitten perceive only verticals, yeah, and a similar result occurred for also horizontal picture. So this result then supports the idea that perception is determined by neurons that fire to specific qualities of a stimulus. And the stimulus is orientation in this case. And in this right-hand figure, it indicates the location of the visual cortex in the human brain, as well as additional areas that are involved in vision. Yeah, and some other areas we'll be discussing later. So the vision areas are part of the vast network of areas that make up about 30% of the cortex, actually. And some of this visual area receive signals directly from the visual cortex. Yeah, and other are parts of sequence of interconnected neurons, some of which are far down the line from the visual cortex. And following Hubel and Wiesel pioneering research at the beginning, other researchers who began exploring this higher level of the visual pathways discovered neurons that respond to stimuli more complex than oriented lines. And then how are complex stimuli then represented by the firing of neurons in the brain? One answer to this question began to emerge in the laboratory of Charles Gross. And Gross experiments here in which he recorded from single neurons in the monkey's temporal lobe. So Gross research team presented a variety of different stimuli to anesthetized monkey. So on a projection screen, like the one you can see in this figure, they presented lines, square, circles, and etc. Some stimuli were light and some dark. And then the result of the research conveys that the firing rate in nerve impulses per second responds to phase, but not to non-phase stimuli. And neurons in the temporal lobe firings to complex stimuli, right? And following that, research by Roca Miranda in 2011 further explained that the neuron in the monkey's temporal lobe are firing in response to phase stimuli, as you can see in the bottom figure here, compared to the non-phase stimuli. So the problem of neural representation for the senses then have been called the problem of sensory coding. Yeah, we're then explaining the sensory codes that refers to how neurons represent various characteristics of the environment. Yeah, so the idea that an object could be represented by the firing of a specialized neuron that responds only to that object is called the specificity coding. This is illustrated in this figure, which shows how a number of neurons responds to 3 different phases, yeah? But only neuron number 4 responds to Bill phase. And only neuron number 9 responds to Mary phase. And neuron number 6 responds to Raphael phase. So notice that neuron 4 do not respond to Mary or Raphael phase, right? It fires only to Bill's phase. Following that, but there is also population coding in the neuron of the brain, or the representation of a particular object by the pattern of firing of a large number of neurons. So according to this idea, Bill's, Mary's, and Raphael's faces are each represented by a different pattern. So an advantage of population coding here is that a large number of stimuli can be represented, but large groups of neurons can create a huge number of different patterns. So there's a good evidence for population coding in the senses and for other cognitive function as well. But some function or large number of neurons aren't necessary. And lastly is the sparse coding. So it occurs when a particular object is represented by a pattern of firing of only small group of neurons, with the majority of neurons remaining silent, as you can see in this figure. So the sparse coding would represent Bill's phase by the pattern of firing of a few neurons, neuron 2, 3, 4, and 7, and different phase also having another pattern of firing. And then recently, neurons also were discovered when recording from temporal lobe of a patient undergoing brain surgery for epilepsy. So basically, it's stimulating and recording from neuron is a common procedure before and during brain surgery, okay? So because it makes it possible to determine the exact layout of particular neuron brains. So this neuron responded to very specific stimuli. So as you can see in this figure, it shows the records for a neuron that responded to a picture of the actor Steve Carell and not to other people. Yeah, it's the experiment conducted by Kuiroka in 2007. However, the researchers who discovered this neuron, as well as other neurons that responded to other people's face, point out that they had only 30 minutes to record from this neuron, and that if more time were available, it is likely that they would have found other phases that will cause this neuron to fire as well. So given the likelihood that even these spatial neurons are likely to fire to more than one stimulus, Kuiroka and her co-workers in 2008 then suggest that their neurons are probably an example of the sparse coding here. All right. And then additionally, so memories are also represented by the firing of the neurons, right? But there is a difference between representation of perception and the representation of memories. Slightly different. So the neural firing associated with experiencing a perception is associated with what is happening as a stimulus is present. And that firing associated with memory is associated with information about the past that have been stored in the brain. Yeah. We know less about the actual form of this stored information for memory, but it is likely that the basic principle of population and sparse coding also operate for memory, with specific memories being represented in the particular patterns, and stored information that result in a particular patterns of nerve firing when we experience the memory. All right, so this is the end of the lecture video of Cognitive Neuroscience Part 1. We will continue the second part of the video. Thank you for your attention, and I'll see you shortly.